Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. I hope I am audible, visible and the presentation right behind me is visible. Please let me know in the chats if I am audible. Awesome. I love the energy in the chat box. Keep the energy till the end of the class. Are we live? Please let me know quickly in the chat box. Okay. Let me check as well. Checking. Yes, we are live, right? Awesome. Yes, awesome. We are live. So today we are going to do microbes in human welfare, which is a very kutti chapter, okay? But this chapter can give you up to 2 questions. But now what is the trickiest part of this chapter is lot of names are there that you have to know and lot of bacteria, fungi, all this you have to keep remembering. So the names are a little difficult and you have to know what is the use of them. See students, whenever we talk about microbes, right, whenever we talk about microbes, we always have a tendency to tell that, oh my god, it's dangerous, right? Microbes always causes diseases, microbes are bad, that, this, always negative about them, right? But the actual fact is microbes are a lot helpful to us in our daily routines, daily things, whatever we do know, those things also microbes are really helpful. So in human health and diseases, I'm going to talk about all the negative microbes, and in microbes in human welfare, we are going to talk about how helpful these microbes are to us in day-to-day life. Okay, so are you all ready? One like for microbes on in the chat, I mean not in the chat box. Smash the like button and mark your attendance because your attendance is very, very important. Okay, yes, some microbes, most of the microbes are useful to us. Let's see which all of them. Before that, 1 second, I cannot see your chats. Chalo, yes. Okay, yes, awesome. So microbes are helpful. Let's see which are the microbes that are helpful. Before that, as I told you, around 2 chapters. So even in 2025, we can expect 1 to 2 questions from it. That means this chapter has a general weightage of 4 to 8 marks. Okay, 4 to 8 marks. This chapter generally has a weightage of 4 to 8 marks. Okay, ready, awesome. Many of you are present, very, very happy. So students, today we are going to do, it's very similar to NCERT line by line because this chapter, otherwise there's no other way to teach it. Okay, it's neither NCERT or I write the entire, NCERT in this board and then teach you. Okay, so I do not want to waste your time because this chapter fetches only like 8 marks. We need to make sure that this chapter finishes in 2 to 2.5 hours, very crisp chapter. Okay, so I've put every word of NCERT, full NCERT is on the board, but I will make you write also with GM Express, that is Gopika Ma'am Express, just like Chennai Express. So students, see, beside the fact that microbes are dangerous, let's also see what are the things that are important to us. So in this first paragraph, they're talking about how beside macroscopic plants and animals, microbes are a major component of the biological systems on this earth, right? And can we see microbes with our naked eye? No ma'am, we cannot see microbes with our naked eye. We can only see it with the help of microscope. Right, we use microscope to see this microscopic organism. So first thing that you have to know about microbes is microbe is a microscopic organisms. Okay, now okay, next thing, microbes are present everywhere in soil, water, air, inside our bodies and of other animals and plants. That means this gives me an understanding that microbes are cosmopolitan in nature, right? Cosmopolitan in nature. That means they are present everywhere. Now, does it mean that they are present only where, like, okay, this condition is very nice, very favorable, I'll be present there? No, they are even present in extreme conditions. Okay, now all of you in the chat box tell me, where did we study microbes? When we did biological classification, we happened to talk about bacteria, we happened to talk about microscopic prokaryotes, unicellular organisms. Where did we talk about? All of you in the chat box. See students, microbes are not present. Like, see, if the condition is favorable, I'll be here. If the condition is not favorable, I'm not going to be there. It's not like that. They are present everywhere. Extreme conditions, like, see, they are present even at the sites where no other life form is seen. Okay, possibly where the geysers, that means hot thermal vents, where the temperature may be up to 100 degrees Celsius, literally like boiling water temperature also, they'll be there. All of you have given me chapter name, biological classification, I already told. Very good, Sai Mona, very good, Manaswini and Madhumati, Monera, right? So students, specifically in Monera, when we spoke about archaebacteria, right, when I spoke about archaebacteria, I told you that archaebacteria, 3 types we studied. What were those? What were those? First we had thermoacidophils, right? Thermoacidophils, that means the ones that can stay in extreme, okay, hot condition, at the same time at extreme acidic condition, okay, extreme acidic condition. Next one was halophils, right? Halophils, that means the ones that were able to stand in extreme salty conditions, right? Salty conditions. Yes, chats are slow for me, yeah? Okay, that could be the reason. Students, you haven't put the target. Quickly put the target. Quickly put the target in the chat box, right? Halophils were there, that is the ones that can stand in halogen. I told you, no, common salt, what is the formula? NaCl. In that Cl is what? A halogen. So halophil means salt loving organisms, right? Then we had methanogens, okay, methanogens, which were present in ruminant animals, right? Gut of ruminant animals like your cattle. Now, if I see here, archaebacteria were the bacteria who were able to stay in extreme conditions, okay? Extreme conditions, okay. Okay, so here students, look here. About 100 degree, okay, 100 degrees Celsius deep in the soil, under the layers of snow, several meters thick, in acidic environment, microbes are diverse protozoa, bacteria, fungi and microscopic animals. Now students, even in fungi, right, even in fungi we have problem causing ones, even in bacteria we have lot of problem causing ones. At the same time, there are also the good ones, okay, good ones. Oh, hi Hansi, hi Hasini, hi to all the students who joined late, came from DNA. Hi. Okay, viroids. Now students, in last class we came across the words virus, viroids and prions, right? These people also are disease causing. Yes or no? These people also are disease causing. Now we have viruses, right? We have bacteriophage, the ones that affects the bacteria, the virus that cause an infection in bacteria, okay? Then we have viroids, where they have free RNA structure, no protein code. Then we have prions, where they have abnormal folded proteins. These all were able to cause one disease, right? Like mad cow disease they were able to cause. So potato spindle fiber disease, all these are also problematic ones. But the thing is, all this while we were studying about problematic one, now let's understand some good ones. So all this we have learned under Monera, okay, under Monera. Is this clear? Introduction is nothing students, just understand that we have learned about this previously, okay, learned about this previously. Okay, now students, look at these images. Are these images clear? Yes. So here if I see, I can see one rod shaped bacteria, yes or no? It's a rod-shaped bacteria. Now, according to biological classification, all of us know that bacteria are of 4 shapes. All of you tell me different shapes of bacteria. One is cocci, okay. One is rod-shaped. One is comma shaped or vibrio, another one is spiralum. Yes or no? These are the 4 shapes, right? Awesome. Okay, awesome. Okay. So here students, the first one was cocci, okay, cocci. Second one was bacillus, that is also called as rod shaped, okay. Third one is vibrio or comma shaped, and fourth one was spiralum, okay, spiral. Same thing here, images have been given. Now all of you look here. The first one that is given is a rod shaped bacteria, which we also call as bacillus, okay. Now example, I can write lactobacillus, okay, from the name itself you know it is rod shaped, okay, lactobacillus, which is the rod shaped one. Here if you see a cocci, that means spherical shaped, spherical shaped. Example, you can think of streptococcus, All this you have studied, okay? It's not like this is nothing is new to you, because in molecular basis also we have studied, right? Streptococcus pneumoniae. Here it is Streptococcus. Students, I will forget to underline, but you should underline, okay? Chalo. Then here, students, actually this rod shaped bacteria, no, they have magnified, and they have shown you this way, so you can see it. Look at this, rod shaped with a flagella, okay, with a flagella. Can you see? Rod shaped with a flagella, okay? So here you can see rod shaped bacteria magnified at 1500x, okay, 1500x. Spherical shape, again magnified, rod shaped, magnified. That rod shaped is magnified around 50,000x, okay, 50,000x. Then only you will be able to see this, otherwise usually we'll see this, okay. Again, Baswa sir would have taught you gram negative, gram positive. Then we use these kind of bacteria only, okay, gram positive, gram negative. So students, from there, here if I see here, I can see a virus here, yes or no? Students, last time from images questions had come, I'm not telling about NEET, like when you're doing mock paper and other things, no, questions come from this. This is a bacteriophage. Now what is a bacteriophage? Ma'am, we know that these people will affect or cause disease in, that means affect the bacteria, okay, affect the bacteria. This bacteriophage here you can see, and hetra, so it can be polyhedral, okay, the head will be polyhedral, it can be of that shape, and they are made up of capsid, okay, the capsomeres, many capsomeres together forms the capsid, the protein code. Inside that, what is going to be there? Your genetic material is going to be there, right? Here, green is not visible, no? Here, your genetic material is going to be there like this, right? Genetic material will be there inside that, okay, and then they have a collar, then they have the tail fibers through which they will attack. Then students, here what can you see? This is an adenovirus. Adenovirus is a virus that has the ability to cause a respiratory disorder. Okay, now during COVID time, all of you know, no, severe acute respiratory disorder. So adenovirus is the virus that has a capability to cause a respiratory disorder. So this image is of an adenovirus. Now if you look here, these looks like those chopsticks, no? Chopsticks. These are your tobacco mosaic virus. This tobacco mosaic virus also is magnified up to from 1 lakh to 1,50,000 magnification is there. Then only you will be able to see this. You know how they look actually? They look something like this, students. One cylindrical shape they'll have, like this, some cylindrical shape, and inside are like this. Okay, they'll be full circled. But if I draw that, it might take 2-3 business days to complete the diagram. So it will look like this. Okay, if I magnify that more, it will look somewhat like this. So this is TMV, tobacco mosaic virus. So this diagram, they're just showing how these diagrams are there, how much magnification we have to do, and what is the situation like. Okay, that is what this is. Okay, chalo. Now when we come to the next diagrams, I have taken all the diagrams, okay, full NCERT is here, so you can blindfoldedly listen to this class and then look at your NCERT and you'll be like, wow, ma'am has put every line of NCERT, okay. So here students, what you're seeing here is 2 Petri plates, okay. Now I don't have a Petri plate, but imagine this is your Petri plate, okay, this tissue paper is the Petri plate. On this Petri plate, I will add a nutrient medium. Now what is a nutrient medium, ma'am? Now when I want to grow a plant or when I want to grow a money plant inside my house, I neither give it water, complete water like on the bottle, right? Nowadays we do a lot of designs and put the water and then grow the money plant, because you know the money plant will grow. Same way, the same money plant, when you are transferring it to a soil, you will make sure that you mix some cow dung or you mix something that is little more fertilizing so that this grows faster. Okay, now same way I put a Petri plate. In that Petri plate I pour some nutrient medium that will make sure that the bacteria or the fungal which I am going to planning to grow will grow. Now this is a liquid medium, right? Now if I pour some water on this is a liquid medium. This is a liquid medium. 1 second. Yeah. Okay. This liquid medium have to be solidified for my agar to, that means for my bacteria to grow. For that I will add some agar. Okay, I'll add some agar. This you would have studied, I think, in plant kingdom. Plant kingdom, there you would have came across the word agar, right? Gelidium, all this agar word would have been. This agar will solidify this entire nutrient medium and make sure that this gets solidified so the growth will happen. Now, I am, let's say Manoj and Paul is there. Both of them did not know that, okay, everywhere bacteria is there, everywhere fungus is there. They know this. But then they were not aware. So what they do is, with taking the Petri plate, they forgot and they kept it open. So when they come after, let's say 8 to 10 hours, next day when they come and check in the lab, they will see growth on that. Now that growth can be bacterial or fungal. Fungus is comparatively less in the air, but bacteria is extremely high. Now if they keep the Petri plate and they have gone, okay, they can see some growth like this, some growth like this. Now imagine that, okay, Rani knew that, okay, bacteria is there. So what she did streaking. This is called as streaking. That means I basically do this, okay. She did a pattern. It could be T streaking, it could be continuous streaking, it could be any kind of streaking, different streaking techniques they will teach you. But this technique, she took something and she started streaking. She cannot see anything on the nutrient medium. Always remember, the agar will be there. She would have taken a streaker. There's one streaker. With that, she would have rubbed like this. After 8 to 10 hours, when she comes back to the lab, she'll be able to see some growth. That will be a bacterial growth, or it could be a fungal growth. Now, fungal growth is very easy to identify. Why? I'll tell you. If this is a nutrient, this is a Petri plate. Okay, this is the Petri plate. And in this Petri plate, I added some nutrient medium, okay, like this agar was added, and I left it open, Immediately, I'll be able to see the next day when I come, I will start seeing fungal growth like this, okay, a greenish color with cottony surface on the outside, okay, cottony surface on the outside. So if I draw an enlarged structure, this is how it's going to look, okay, like this, and they will have some cottony structures like this, This is very important for you to understand, okay. Should know how this growth has happened. Now same thing, let's say Meghana took the nutrient medium or the Petri plate and she went into the restroom, near washroom area she went, or she went to a place where there was a lot of garbage. She just have to keep it open and then close it. And students, you won't believe, next day the entire Petri plate will have lot of fungal and bacterial growth. So that means from this, what do you understand? That everywhere in the air, every single place, there is always some amount of bacteria, fungus, and they will immediately start growing in this nutrient medium. Okay. So this is with the help of streaking. You can see here some streaks they have made. Then everywhere dots have happened. Okay. Everywhere dots have happened. Can you see? See, some places, this looks like, okay, the person did not plan it. Randomly, they touched it by mistake. And here they can see that some streaking has been done. Here you can see a proper fungal colony. Okay. So they form colonies. Okay. They form colonies. Visual colonies, that means you will be able to visualize it, right? Starting, you did not see anything. When Meghana started it, when Paul started it, they did not see anything, but they just touched something and they started doing the same thing. But later on, they started noticing, oh my god, did I actually put this much bacteria, or did I touch into this much bacteria? That is how it is. But it is a very beautiful, like microbiology itself, you have to be careful, but it is an amazing thing. Okay, it's an amazing thing. You'll be amazed with how beautiful things are there, but it's scary as well, because if you leave it open, you have a chance of contaminating every other Petri plate in the lab. Okay, every other Petri plate in the lab will get contaminated if you leave it open. You have a fungal growth inside and you left it open. You did not close it. They have spores, right? So because that spores will fall, it will get scattered everywhere. So you have to be very careful. Now, microbes in household. So students, this chapter is divided into household, industrial, then you have sewage, biogas, then biocontrol, then that. Okay, very easy BC. Okay, lemon squeezy chapter. Now look here. You would be surprised to know that we use microbes or products derived from them every day. Okay, we understood. Now look here. Microorganisms such as lactobacillus and other commonly called as lactic acid bacteria, which is called as lab, grow in milk and convert it into curd. Now, as I told you, you have boiled some milk. Okay, this is boiled milk. To this milk, you would have seen your mom adding 1 spoon of curd. Okay, 1 spoon of curd. Got it? That 1 spoon of curd, students. Okay, this is the most fanciest spoon you can see. That 1 spoon of curd that your mom added is going to act like an inoculum. It's going to act like a inoculum. That means what? This 1 spoon of curd has millions of LAB, lactic acid bacteria, or your lactobacillus. Now what are they going to do? Okay, what are they going to do? They are going to go into the milk and convert all this lactose into lactic acid and convert this milk into curd by tomorrow morning. Okay, you will have fresh home prepared curd. Now, how does this happen? This happens because of one and only bacteria called as your lactobacillus, which is a very, very useful bacteria. Okay, got it, students? Now, curd is more, curd is always better than milk because of the presence of vitamin B12. It's very helpful, okay? So, that's why they tell you should consume lot of curd. Probiotics are there and other things, Greek yogurt and all that. So, like that, probiotics are more. Compared to milk, curd is more better, okay? So, is this clear? So, the first ever useful bacteria that you have to study is lactobacillus, which will convert your boiled milk to curd with the help of LAB or lactobacillus, that is lactic acid conversion, okay? Where all the lactose in the milk will be converted to lactic acid and then it will become curd. Now, the inoculum means the curd that you added, that 1 spoon curd that you added to the boiled milk to make sure that this get converted, because something should be there, something should be there to activate the whole process. That activation is done by this. Clear? Chalo. Now, this is all you have to remember. Now, a small amount of curd added to the fresh milk is as inoculum or as starter. We can use these two words, but generally we use the word inoculum. We do not use starter, right? When you think about starter, you think about non-wet starter, wet starter. So, you use the word inoculum. Contains 1 million of lab, which are suitable temperature. Now, what's the suitable temperature? Did your mom tell you to check the temperature, students, anytime? Okay. Anytime has she told you to, what happened, Sony? Current went, power cut. Students, target is not reached, yeah? Where is the attendance? Or lazy people, morning they wake up, open the this one and they go to sleep. Yes. So, students, now has your mom checked the temperature in a thermometer? Oh, I'm keeping the milk at this temperature. Nothing like that. She makes sure that the milk is boiled and then becomes warm. But generally, if you want to make it in a larger scale, we always check the temperature. It should be favorable. That is 40 to 42 degrees Celsius is a favorable temperature for converting milk to curd, okay? For converting milk to curd. Thus, converting milk to curd, which also improves its nutritional quality by increasing vitamin B12, okay? In our stomach too, the lab plays a very beneficial role in checking disease-causing microbes. That's why they tell that, okay, whenever even nowadays if you see health as a whole concept, people tell you have a lot of Greek yogurt, have a lot of that, this and all, no? That's mainly because of it, because it makes sure that the gut health is very important, okay? Gut health is maintained. Students, this is clear? We will write all of this in GM Express, but right now try to understand, okay? That is more important. Understanding is more important. I will write it. I know you people love seeing me write, but wait for some time. Okay, now, the next one. The dough which is used for making food as is dosa and idli are also fermented by bacteria. All of you would have seen this. Yes, all of you would have seen this. Full active. They will be active when your results come, right? So, students, this is dosa and idli is because of leuconostoc. Okay, this is the bacteria that will actually ferment your idli and dosa. So, tomorrow you have to tell your mom, Mama, did you add leuconostoc or is there leuconostoc in dosa and idli? Then there will be a very easy exit from the house. So, all the best, please do it and let me know what has happened in your house, okay? Got it? Don't miss it, okay? Leuconostoc, that is the bacteria that will make sure that dosa and idli is getting fermented. Tomorrow, without fail, ask your mom, or after the class, ask your mom, is there leuconostoc in whatever you have prepared? If it is not there, then sorry, tomorrow idli will not be served. Okay, tell like this and see your mom's reaction. Okay, now, the puffed up appearance. Students, how does this idli, dosa, the puffing happens? Because of the presence of carbon dioxide, okay? Production of carbon dioxide. Even your bread, everything, same principle will be used, okay? Cheese, everything, okay? Production of carbon dioxide is the main reason how your idli gets puffy, dosa gets puffy, whatever, it gets puffy, okay? Now, can you tell which metabolic pathway is taking place in the formation of carbon dioxide? You have to go to your anaerobic and aerobic respiration, no? There you would have studied, right? Fermentation, alcoholic fermentation, that is what they're talking about. Now, look here. Similarly, the dough which is used for making bread, okay? Bread is also fermented using baker's yeast. So, yeast, same yeast you are going to learn 2 times, okay? Same yeast, that is I think here somewhere it should be written, no? Yeah, here it is written, okay. Saccharomyces cerevisiae. So this one has two functions, okay. One, it will be used as baker's yeast, that means to make all the bread and everything, right? Baker's yeast, okay, baker's yeast. And second one, the same yeast will be used as brewer's yeast, okay, brewer's yeast. That means to make your beer, wine and all of that, no, brewer's yeast will be used. Is this clear? Okay. Did you already get? Tomorrow you should get. Today you ask. Okay, tomorrow you get, or in the night you ask. Is there lactobacillus in this? Can I consume? Is my vitamin B12 going to increase? Otherwise, I really don't want your curd. Like this you tell and let me know your reaction, because it will be fun, students. Sometimes we need fun, no? It's okay. What is there in that? Okay, it's your mom. She'll just throw some things on you. Okay, you should be in a position to attack and make sure that you don't get any hittings. Okay, this is Katron ki Khiladi. Okay, yeah. So Saccharomyces cerevisiae is the one that is going to act like your, look here, baker's yeast as well as brewer's yeast. Baker's yeast obviously to bake. Okay. Right now, a number of traditional drinks and food are also made by fermentation by microbes. Example is toddy. Students, toddy is very famous in Kerala. I got to know that it is also famous in Andhra Pradesh and Tamil Nadu as well. Toddy is a alcoholic drink which has an alcohol percentage of 3 to 6%, I believe. So this alcoholic drink is made from palm. We usually see it with coconut tree. I think that is the Misho product of toddy, and the actual product is made up of palm. Okay, Caryota, urines, and also many other palm names are there. Different places, different palms they use. This one will be kept, and they will extract the sap and they will ferment it. Okay, and then they will be giving it for consumption. After fermentation, if in case the fermentation exceed, like in the sense toddy is not sold and then it kept fermenting, then it can be also used as vinegar because it gets very bitter. Okay, is this clear? So toddy is a traditional drink in some parts of southern India which is made by fermentation of sap, okay, from palms. Okay, from palms. Now, microbes are also used to ferment fish, soybean and bamboo shoots to make food. Cheese. So first, what and all did we study? Curd, milk, clear? Yes or no? Are you all following me? Hello, back to class, all of you. All of you back to class. Something they're talking. Okay, yeah, Telangana also it's special, right? Okay, so students, first we spoke about dosa and idli, which here bacteria name is not mentioned, but I have mentioned your leuconostoc, which is helping in your fermentation, and the puffiness is because of the production of carbon dioxide. Okay, same thing is going to happen in cheese also. The holes in the cheese, no, the bigger the holes, that means more production of carbon dioxide is there. Okay, now where do you think? After that we have come to bread. Bread is made by your yeast which is called as Saccharomyces cerevisiae, which will act as a baker's yeast at the same time as brewer's yeast to help in making beer and wine. Okay ma'am, understood. From there we came to toddy. About toddy you don't have anything to learn, you just have to know toddy also is a fermented drink which is fermented from sap of palm. The palm can be Caryota urines or it can be any other palm. Many palms are there if you just check. Okay, okay, you can check the different types of palm that are there. Now, they are telling that microbes are also used to ferment fish, soybean and bamboo shoots to make food. Okay, to make food. Now, cheese. After that, we are going to cheese, very important. Cheese is one of the oldest food items in which microbes were used. We know Tom and Jerry, right? Tom and Jerry is like always the epic cartoon ever to exist. So Tom and Jerry, the whole series runs on the cheese, right? So here, look here, different varieties of cheese are known by, very important. How do I categorize cheese? How did I know, right? This was okay, cheddar cheese, cottage cheese, etc, etc. Many cheeses are there now. How did I know it? It is because of I observed the texture, the flavor and the taste. Basis of this three, we categorize the cheese. Is this good? Okay. Carbon dioxide, Sony, due to carbon dioxide, because this puffiness, the holes, in the cheese also, that is because of the production of carbon dioxide. How much it has got fermented, okay? So that carbon dioxide release makes that. Now look here, the speciality coming from microbes used. For example, the large holes in Swiss cheese are due to the production of large amount of carbon dioxide. Is this clear to all of you? So you have to remember two things about carbon dioxide, that is the puffiness, okay? Puffiness in your bread and the holes, large holes in your cheese is depending on the production of carbon dioxide. Is this clear? Okay, it's because of carbon dioxide. Yes, I think Sony's answer. What? What is that? What are you telling? I think Harshain woke up in the wrong side today, students. Okay. Kuch bhi. Okay, now large amount of carbon dioxide by a bacterium named Propionibacterium shermani. Students, this is a very shy bacterium. Shermani, I think in Hindi, no? Okay. So, look here. Propionibacterium shermani is the bacterium. See, that is Swiss cheese are due to the production of large amount of carbon dioxide by a bacterium named Propionibacterium shermani. So, Swiss cheese shermani. Okay, Propionibacterium shermani. Now, look here. The Roquefort cheese are ripened by growing a special fungi on them. Okay, special fungi on them. Is this clear? Special fungi on them. Are you all following or laughing for Harshain's? What happened, Madhumati? Yeah, exactly. Likes are not coming. Okay, very conjuice people are in this world. Is this clear? Any doubts? If any doubts, ask me right now. Swiss cheese clear? We'll write this, but still, I'm just asking you people. Okay, chalo. Okay, so Roquefort cheese are ripened by growing a specific fungi on them, which gives them a particular flavor. Okay, particular flavor. So this is how much, I mean flavor, not favor, particular flavor. Now, microbes in household, let's write it. Okay, this is the part of your GM Express itself, okay? Okay, this ads are coming, huh? Can't do anything, students. Even for me, it's coming. Imagine for my class, for me only it's coming. Okay, so first we start with lactobacillus, okay? This one slide will be enough for you to study the whole thing. So cooperate and let's make notes. Lactobacillus. What is it going to do? It is going to convert milk to curd, okay? Milk to curd. Now students, the curd that you add, right, the curd, okay, the curd that you add is going to act as an inoculum, okay? It's going to act as an inoculum. I'm just writing it here. It needs around 40 to 42 degrees Celsius for milk to curd conversion, okay? Fine. Now, next one is, which is next one? Dosa idli, I'm writing it here, even though it's not in NCERT. Dosa idli, as South Indian, if you don't write this, it will be disrespect. Okay, this is leuconostoc. Leuconostoc. Chalo. Next one. Quickly, chat should keep moving. Okay, next one should already be there. Yes, ma'am, this one. That I can't see. That I can't see. All lazy people. Okay, next is cheese. Right? Not cheese, bakers. Okay? I should have written this one this side, no? So in 1 second, we'll write all the bacteriums one side and the purpose the other side, otherwise it's going to be too confusing. Give me 1 minute. Since tomorrow also I'm taking class, okay? Don't forget, I know tomorrow is Sunday, but we have to take class. That is, tomorrow I'll be taking reproductive health. Okay, reproductive health. Okay. Exactly, Diksha ma'am's favorite food. We have to write with respect to Diksha ma'am. Okay, leuconostoc. Yes. Now this fellow is going to convert dosa ma'am's dosa and idli. Okay. Students, next class ask ma'am. Okay. Ask question about leuconostoc. Got it? Ask ma'am about leuconostoc. If ma'am doesn't know, then we will not allow ma'am to eat any more dosas. Chalo. Then she'll ask me chemistry question back. So don't be very sweet. Okay. Next one is yeast. Saccharomyces cerevisiae. Okay, cerevisiae. One H is here. This fellow has two things. I'm writing both, okay? I'm writing both, even though again alcohol is a separate session by itself because it's industrial. It's coming under industrial. So right now I'm writing baker's yeast. Okay, baker's yeast, and the puffiness. Okay, the puffiness in bread is because of the production of carbon dioxide. Chalo, done? Hey ma, why didn't you listen to the class? Inoculum is something that you add into the milk because there should be someone, no? LAB, from where are you going to get lactobacillus? Lactic acid bacteria, from where are you going to get? That you will get it from the curd. So that 1 spoon of curd, we will add it into the milk because that will have millions of LAB, which will convert all the lactose in the milk to lactic acid. Okay, got it? Chalo. Yes. Brewer's yeast, we'll write it under industrial. Okay, we'll write it under industrial. Saccharomyces cerevisiae done. Then we will go directly into toddy. See, toddy, I'm not going to write any bacterium name, but I will write Caryota urens is the palm that is used. Okay, the palm that is used to make toddy. Okay, to make toddy. It's an alcoholic drink. Okay, it has some percent of alcohol. Next one is cheese. Swiss cheese is given. Swiss cheese is given because of propionium. Okay, propionibacterium shermani. Okay, done? Yes, Propionium bacterium shermani. Then we have Roquefort cheese. Okay, Roquefort cheese. This is by Penicillium Roquefort. Okay, Penicillium okay, Roquefort. Students, again I wrote this side. Ayyo, why is Gopika Ma'am like this? Kallu. Even in Malayalam it's Kallu only. Okay, Kallu. Tell la. Okay, we tell Kallu. Yes. Got it? Okay, done. Okay, so Swiss cheese and Roquefort cheese is Propionium wait, students. Okay. Propionium bacterium shermani and Penicillium Roquefort. This is all about it. Did we miss something? Okay, did we miss something? Kalloli, yeah, even in Tamil also it's Kalloli. Okay, fine. Okay, such a common language, common word for one thing. Okay, students, this is clear. This is all your household bacteria. Now we are going to the industrial ones. Okay, industrial ones. So now look here. Industrial ones, you mainly have about beverages, that is the alcohols. Okay, even in industries, microbes are used to synthesize a number of products valuable to human beings, especially your beverages and antibiotics are some examples. Production on an industrial scale requires growing microbes in a large vessel, okay, called as fermenters. So students, we are doing, see, now we were talking about small things, right? In a glass you take some milk and you convert it to curd. It's a very small thing, right? Industrial thing. So here we are talking about larger things. So we need something called as fermenters to make sure to grow all the microbes inside this. Okay, to do any conversion we have to grow them in large fermenters. Got it? Yes? What happened? Okay, clear? Chalo. Now, main portion. Okay, main portion is about your beverages. Okay, look here. Microbes, especially yeast, have been used from time immemorial for the production of beverages like your wine, beer, rum, whiskey, gin, etc., etc., etc. Now, anyone drink? I will give you people one. Now I'll call your parents. Okay, what question you are asking in the chat box? As if you are taking votes to go to some place. Okay, so here students, there's a thing. Yes ma'am, Saccharomyces cerevisiae is a brewer's yeast which can ferment and form the alcohol. But there's a problem. This yeast will commit suicide. That means the yeast will kill itself if the alcohol percentage exceeds 13%. That means if the alcohol, okay, percentage exceeds 13%, then this yeast will not be of any use because the yeast will die. Students, it will kill itself, students. Okay, yeast will die. Now, ma'am, okay, fine. Now, in beer, okay, in beer, the alcohol content is around 3 to 6%. So, I can definitely ferment beer with yeast because 13%, right? Wine also, the alcohol percent is around 9% to 12%. This also I can ferment. But what about the ones which have 40 to 60% alcohol? What about the ones who have up to 70, even 90% absolute alcohol? How do I ferment that? For that, I use a process called as distillation. So you have distillated drinks and non-distillated drinks. Now, all of you, yes, exactly. Abhi, very nice. Okay, so this one, oh no, this got stuck, students. Give me 1 minute. Okay, oh. Where did it going? Chalo. Okay, board got alcoholic feeling. Okay, board got suddenly excited. So here what is happening? This will become non-distillated. Okay, non-distillated. Because I can ferment it with the help of yeast. I do not need distillation here. Okay, I do not need distillation here. Got it? But the other ones, I need distillation. Who are those? Who are those? All of you in the chat box. Those are your rum, okay, whiskey, vodka, gin, okay, absolute alcohol, etc., etc., many other. All this will have extreme alcohol level. Let's say I'm just keeping an approximate, okay, 40 to 60%, okay, 40 to 60%. So this one I am going to distill, okay, this will become distillated drinks, okay, distillated. Clear? This is one of the industrial use. Done? Done? Brandy, that also I forgot, students. Rum, whiskey, vodka, brandy, okay, that I forgot. Okay, chalo. Now look here, Saccharomyces cerevisiae used for making bread, okay, and commonly called brewer's yeast. Okay, commonly called brewer's yeast is used for fermenting malted cereals and fruit juices. Now, example, if I look here, wine is actually fermentation of fruit juice. But if I look at the beer, it is malt cereals. Okay, so that is wheat, students, wheat. Okay, wheat. Very interesting. My students are in next level of happiness because I am talking about these things. Okay, full excitement. All the boys in the chat box are like, yes ma'am, very nice topic. I really like to understand about this topic. I'll give you one. Okay, only understand about the topic, nothing more than that. Okay, nothing more than that. Okay, yes. So it is used to ferment malted cereals and fruit juices to produce ethanol. To produce ethanol. Got it? Now, do you recollect? Do you recollect? Please tell me the answer. Okay, metabolic reaction which results in the production of ethanol by yeast. You all know alcoholic fermentation, ethanol will be formed, right? That's why if you keep something with a lack of oxygen, you will start getting that alcoholic smell. Okay, now depending on the type of the raw material used for fermentation and the type of processing, that means with or without distillation, different types of alcoholic drinks are obtained. Like wine and beer are without a distillation. Okay, rest everything are with the help of distillation. You understood what is the reason behind it? You understood what is the reason behind it? Is because yeast will commit suicide, students. Okay, yeast will commit suicide. Okay, and it will commit suicide when the alcoholic percentage reaches up to 13%. Then how do I make the rest of the drinks, right? Not me, other people. So that is why. Got it? Huh. Yeah. Understood. Okay, fermenting plants. Is this clear? Any doubts? You want to know about each of this? You have to search. Okay, NEET may question will not come. Okay, NEET question will not come. What is it made up of and all that? Exactly. I was astrologer before, no? That's how I know all of you so well. Okay, students, beer is with malt. Okay, barley, wheat, all that kinds of beers are there. Now, wine is because of fruit juices. Whiskey is again fermented cereals. What is it? Gin, no? Vodka. Vodka is because of starch. Okay. Brandy, fruit juices. To tell it is very nice, but don't consume. Okay, next one. Antibiotics. Okay, antibiotics. Antibiotics means what? Anything that will attack my foreign particle. One foreign particle has got into my body, I will call it an antigen. Anything that attacks an antigen. Okay. Now students, antibiotics, the first ever thing that came was because of Alexander Fleming. He came up with penicillium, but again, that was also by fluke, right? How you people think of waking up in the morning at 9 o'clock and starting your preparation, but by fluke, you wake up at 11 o'clock and then you change your entire plan, no? So everything happens by fluke even in this discoveries, but their names came in textbook, your name should come in the board. Okay, now look here. Antibiotics produced by microbes are regarded as one of the most significant discoveries. See, even though beverages are there, they are of industrial use, antibiotics was something that actually changed the revolution because Chain and Florey, two scientists called as Chain and Florey, used this antibiotic concept during the World War and helped a lot of people. Got it? Saved a lot of people, helped a lot of people. So that's why this was very revolutionary. Okay? Giving alcohol, can they help anyone? No. So that's why this is very revolutionary. Got it? So here, discoveries of the 20th century and have greatly contributed towards the welfare of human society. This is what they are talking about. Now look here. Anti is a Greek word, means against, and bio means life. So against life, that means a foreign particle who has life, against life. Got it? So antibiotic, antibiotic. For the longest time when I was making this alarm of DNA batch, I every time forget to switch off. Okay, I have to switch it off. Yeah. So students, this antibiotics is against life. Okay, against life. Yes. Now look here. How did this antibiotic discovery happen? That is what they are talking about here, students. So whereas with reference to human beings, there are pro-life and not against, right? It's not against life. We are not killing the useful ones. We are actually killing the unnecessary ones. So it is pro-life, okay, pro-life and not against. Antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other. It's not necessary that every time it has to kill. Sometimes it can also control, it can stop the growth also. Now example, you are affected by rhinovirus, that means you have common cold. You take an antibiotic, the antibiotic is going to reduce, right, the effect of sneezing, and that is because your rhinovirus has been reduced. It's not like we can completely eradicate it, because it will come back again, because multiple variants are formed. So sometimes it is to reduce as well. Now look here, you're familiar with commonly used antibiotic which is called as penicillium. Now here they're talking about the discovery of penicillium. So students, what happened? No, let's say this was, let's go to Alexander Fleming's lab. Okay, we're going to Alexander Fleming's lab. Yeah, Shri, okay, that has to change. It's very disheartening. So this was a Petri plate, okay, Alexander Fleming kept. Students, see, I'll tell you one thing. In life, we have to have power. We have to see, only if we have power. And right now, the entire world runs on a simple piece of paper called money. Okay, you have to have power and money to win such situations, and it is a sad reality. If justice has to be served, then you need to have some power, you need to have some money. So that's why I'm telling you, try to be, if you become a doctor, try to be the most powerful doctor. Okay, try to be a doctor that everyone talks about. Otherwise, don't be. Because if you don't have that power, if you don't have that voice in you, students, we have to keep crying every day for our own justice, for our own safety. You have to, if you want to be powerful, then you have to sometimes step back, sometimes calm down, sometimes not talk, sometimes not react. Okay, some things you have to ignore to become very powerful, because once you're powerful, you realize that, okay, wait, now I have the power to change the rules. Until that, you can only complain about the rules. Okay, now we can only complain about the rules because we don't have the power. But once we are powerful, we can change the rules. So that should be your motto, that I will be so powerful that I will change the rules one day. Imagine if you were the principal of that college, or imagine you were in a position that you could change something, then the effect would be different. Now we can only stand and feel bad and pray for her, but so that's it. Okay, okay, so look here, students, here we are going to Alexander Fleming's lab. Okay, this is the Petri plate he used. This is one more Petri plate he observed. Okay, 2 Petri plates were kept. So students, starting, as I told you, initially you put a nutrient medium. Okay, nutrient medium, and that nutrient medium, on the nutrient medium some bacterial growth happened. Okay, I'm drawing it with orange color to show you people. Okay, some bacterial growth happened. Okay, fine. Okay, first day he kept it, he used a nutrient medium and he kept it. Later, after 8 to 10 hours, he noticed that there was some growth here. Okay, there was some growth here. After that, okay, after that, he noticed that in few places, can you see the pattern change? Okay, students, can you see the pattern change? He noticed that something was growing here. Okay, something was growing here that was stopping the growth of bacteria in that place. Okay, that is when he realized there is something that goes against life. But this was by fluke. Fluke means by mistake. Okay, he did not know it. He did not wantedly do it. It just happened. Now let's see what is the meaning of the story here. Look here. So is this clear? This is how his Petri plate looked first. Later it looked like this. That is, wherever the penicillium was growing, he noticed that the bacteria was getting inhibited, or the growth was not happening. So look here. This, what they're trying to say is here. Alexander Fleming, okay, Alexander Fleming, while working on staphylococci, okay, staphylococci or staphylococcus bacteria, once observed a mold growing in one of his unwashed culture plates. That's what I told you, it is by fluke. Okay, he did not knowingly happen. So that means in one of the unwashed culture plates around which staphylococci could not grow. So he used this. He forgot to wash it, and he kept this unwashed plate in the lab. First of all, you're not supposed to do that, okay? But if Alexander Fleming was there, I would have told. But you're not supposed to do that, okay? Not supposed to leave any petri plates open or unwashed. Everything has to be sterilized, okay, in a microbiology lab. So look here, this was unwashed and he left it there, and he noticed that in one place there is a mold growing, okay? This is the mold that I am showing, okay? Mold that was growing. This mold did not let the growth of Staphylococcus. So what I have drawn here is Staphylococcus. Cocci. Okay, Staphylococci. Okay. Now, he found out that it is due to a chemical produced by the mold named as Penicillium notatum. Okay, after the mold, Penicillium notatum. So, the entire mold was called Penicillium notatum. Okay, and that was the penicillium was the one that was released. Now look here, however, its full potential as an effective antibiotic was established much later by Chain and Florey. That's what I'm telling you. He just knew that, okay, something was there. Okay, something was there. It's not visible. What is not visible? Okay. What is not visible? Hey, slide is visible now. Okay, so here, after this was the finding of Penicillium notatum. Okay. Penicillium notatum. But later, who gave a confirmation? Chain and Florey. Okay, Chain and Florey used this during the World War, okay, to help a lot of soldiers. So this antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain, Florey were awarded Nobel Peace Prize in 1945, 2 years before the independence of 1947. No, so 1945, Chain. So these three people got Nobel Prize for finding this out, because it was also helping the mankind. Got it, students? Very simple. These two people gave the confirmation. Clear, no? Very easy peasy. Okay, Penicillium notatum. That's all you have to remember. Now, after penicillium, other antibiotics were purified from other microbes. And can you name some other antibiotic and find out their source? Please do that, okay? Homework. And antibiotics have greatly improved our capacity to treat deadly diseases, right? Plague, okay, whooping cough and diphtheria. All of this has been treated lately because of antibiotics, okay? Why, students? Nowadays ads are more, or before also it was the same? Or YouTube has increased it? Drink water, all of you. So treat deadly plague, whooping cough, okay, diphtheria and leprosy. All this was recovered. All this recovery has happened because of antibiotics. And can you imagine a world without antibiotics? Tell no. No to NCERT, okay? Yes. I think it has become more so that more people take premium. Like everything happens like that, no? It's business. It's business, because your irritation level increases, and when you get irritated, you automatically be like, okay, it's okay, 100 rupees, what is going to happen? 120 rupees, what is going to happen? And you end up buying it. So it's smart idea, students. I don't know what and all I've taught in this class. Other than microbes, I've taught you about life classes. Now I'm teaching you about what an MBA student should be telling you. So yes, okay, now look here, chemicals, enzymes and other bioactive molecules. We have this, students, I'll tell you entire microbes. No, I have checked NCERT questions, and this question though is like NTA's favorite question, okay? NTA is like, whatever happens, this one question will come from microbes. That is Aspergillus niger, Clostridium butyric acid. This question has come every time. How many of you agree to this? Yes or no? Those who have solved previous year 5 question papers also, you know one question like pakka. Okay, pakka. Okay, that is this question. Look here. Microbes are used for commercial and industrial production of certain chemicals like organic acids, alcohols, enzymes. Now, example. First one, Aspergillus niger. Okay, it's a fungus, students, which forms citric acid. Okay, Acetobacter. Acetobacter will be a bacterium which forms acetic acid. See, I'll tell you, only learn niger. Okay, only remember niger. Rest everything you will be able to relate. I'll tell you, see, Acetobacter acetyl, that means forms acetic acid. Clostridium butylatum forms butyric acid. Okay, lactobacillus forms lactic acid. Only thing that has no connection with each other is Aspergillus niger, that forms citric acid. So remember this one thing, rest everything you'll be able to remember. But I'll tell you one tricky thing that NTA does is, they will give only Clostridium. They'll write Clostridium B dot, or they will write Aspergillus species. Okay, like this they'll write. So that time it can be a little confusing. Okay, so look here, this is the favorite question of NTA. Okay, NTA's favorite question this is. I'll write it that side. Now look here, yeast Saccharomyces cerevisiae is used for commercial production of ethanol. Microbes are used for the production of enzymes. Now next, enzymes. Okay, lipases, students, lipases are used in detergent formulation. Okay, detergent formulation. Why lipases are used? All of you know hydrophobic, hydrophilic, right? So how does this work is, they form a micelles. Okay, they form a micelle. That means when I am applying soap, there is dirt there, that they will all the hydrophobic tail and the hydrophilic head, they will move and they will form a circle. This way what will happen, the oil, the dirt from that place will be removed. 10th standard we have learned this, right, in one of the chapters. So that you have to remember here. I think in your carbon and compounds or one of the chemistry chapters this is there. Okay, chemistry chapters this is there, and are helpful in removing oily stains from the laundry. You must have noticed that bottled fruit juices brought from the market are clearer. How many of you have noticed? You know, you buy fruity, at the same time you get a mango milkshake. Okay, mango milkshake. You will see that in this, you will see some fibers, right? You will see some fibers which will get stuck in between your teeth. You will find that some you know, like grains will be there. That is because in your bottled shampoo, okay, in your bottled, why am I telling shampoo? Drink, okay, juice, it will be clarified. Okay, pure it will be. Appy Fizz, students, I cannot even tell you. Apple juice and Appy Fizz have like two extremes there. One is north, one is south. Okay, so that is because they have clearers, okay, used compared to those that made at home. This is because the bottled juices are clarified with the use of pectinase. Okay, pectinase and protease. Pectinase and protease. Now streptokinase produced by bacterium streptococcus, that will come to. Okay, that will come to. sorry, shampoo. Okay. Oh, that chapter is deleted, huh? Yeah, me and tongue twist is the general thing. Okay. Okay. I think I'll become famous because of my tongue twisting. Students, because so many terminologies, yeah, normal it is to get tongue twist. Otherwise, I have to slow down. More, I have to slow down. Like when slow, this I have to go. So that's why I'm not doing that. I think I've added a slide here. Let's see. After this, there's a slide. Okay. Now, streptokinase produced by bacterium streptococcus and modified by genetic engineering is used as a clot buster. So, students, what happens is, in case in your heart there is clot. Okay. Rasna, if I tell you when I'm coming back to DNA batch, you will have tears in your eyes because it is going to be a long break. Okay, I'll come next month. Next month last I'll come. Okay, so yeah, till that you have to see me in RNA batch. So students, what is happening is, clot buster means, imagine in your heart there was a clot. Now, when the heart is trying to pump blood, it will try to push the clot, right? So that time your heart will start beating very fast. So that, like, imagine someone is standing and you're trying to push them. Obviously, you will put all your pressure to push them, right? So that will make you, the entire system will start boiling because of that pressure. Oh, this person is not moving. Same way, the heart also will try to pump as much as blood, the pressure, to increase the pressure so the clot moves. But that will lead to the clot burst opening and blocking the entire pathway. Now imagine an artery is only blocked here. Here there was a block. Now this will burst and spread everywhere. I have taught you this in body fluids and circulation. So instead of that, we will give streptokinase, which will dissolve the clot. Okay, dissolve the clot. Got it, all of you? Yes, yes, removes clot or dissolve the clot, okay? Yes, yes, I'll miss you people, I'll see you people here, okay? For removing clots from the blood vessel of the patient who have undergone myocardial infarction leading to heart attack, got it? Now, another bioactive molecule is cyclosporin A. Students, all of you know cyclosporin A, what is it? It's an immunosuppressive agent. I have told you before also, if you sell your kidney to buy iPhone or you sell your kidney to buy PS5, that means at that time you sold your kidney. Now you want one kidney back, okay, or you sold both of your kidney. Then you want a kidney back, right? You will take it from your relative or someone close to you. That time your body shouldn't reject it. That is, graft rejection happens, no? To avoid graft rejection, we give immunosuppressant. That means to suppress the whole reaction. That is called as cyclosporin A, okay, which is extracted from here it is there, no? Produced by Trichoderma polysporum, okay, Trichoderma polysporum. From this question also has come. Okay, neither they'll give you Aspergillus niger, they'll give you Trichoderma polysporum, they'll give you Monascus purpuris, and one more they'll give you, anything they can give you. Got it? Chalo. Now look here, statins produced by the yeast Monascus purpuris have been commercially used as blood cholesterol lowering agent. Students, this is about bad cholesterol, okay, bad cholesterol, not the good one, okay. We need some amount of cholesterol. Nowadays people show no cholesterol at all, no fat at all. That's not true. You need to have 40 to 50% is your muscle, rest have to be other things, no. So fat should be there, cholesterol should be there, good cholesterol should be there, not the bad one. That bad cholesterol you have to burn it out with the help of exercise, but do not believe the fact that, oh, no fat, zero fat. Okay, that is all unnecessary. This one, blood cholesterol lowering agent, that is called as statins, which are taken out from Monascus purpuris. All this you have to know. There's nothing to teach. It is all names that you have to know. Okay, it acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. Here your enzyme inhibiting complex comes. Okay, biomolecules you would have studied, no? Are we ready to write? Who wants break? My tongue needs break. Always. Okay. Because I don't talk other times this much. So when I talk suddenly so much, my tongue is like, why? So students, Aspergillus niger will form your citric acid. Okay. Citric acid. Okay. Next one. I think space should be enough. Yes. Tell me one by one, all of you, fast. Okay. Whoever repeats will be out. Okay. None of these are straight lines. That's the speciality. Okay. Next one. Aspergillus niger, done. Next one. Acetobacter. Acetobacter, acetyl. From the term itself, you know, it is acetic acid, which we commonly call as vinegar. Okay. Concentration difference will be there if you want to use it in lab. Otherwise, normal one is vinegar. Okay. Yes. Next one, Clostridium. Students, I am not underlining. Okay. Don't tell later in the comments, ma'am, you did not underline. Okay, butyrium, Clostridium butyrium, that is your butyric acid. Last one, lactobacillus, okay. Lactobacillus, that is forming lactic acid. Acid. Okay, done. Now this side we are going to write all the rest of it, okay, rest of the enzymes and other things. Okay, first one we will write streptokinase. What is streptokinase going to do? It will act as a clot buster, okay. Don't write blockbuster, okay, clot buster. Blockbuster is movies. Okay, streptokinase is clot buster extracted from streptococcus, that you please make a note. Now next one, okay, I remember cyclosporin, so we'll write cyclosporin, okay, A, that is immunosuppressant. Students, all these notes you will get on Vedantu NEET English Telegram, and if you want to join my Telegram, it's Biology by Gopika. Okay, got it? If you people don't comment, you know, you people are not commenting nowadays. I see that. Okay, I see that. How much time does it take to tell thank you ma'am, class was good? How much time does it take? People sit and comment on everyone's photo, no? That half of that time won't take. Okay? They will not even respond to you. They'll put one heart and go. At least me, I'll smile, I'll appreciate you later. If you people don't, if I don't see all these names today, no, then you see people. Okay? Then you people will get cyclosporin A, immunosuppressant. Okay, I'm joking. Which was extracted, cyclosporin A, immunosuppressant. Yeah, it's extracted from what, students? Trichoderma. This is the most important, okay? Knowing it's an immunosuppressant is also important, but you have to know this. This is more important. Trichoderma polysporum. See, 2 times if you study this chapter, more than enough. Okay? You will remember it forever. Cyclosporin done. Next one is statins. Okay, these are cholesterol lowering agent. Cholesterol lowering agent. This is extracted from Monoscus purpuris. Done? Okay. Yes. Did we miss something? Did we miss something or did we do everything? We did not miss anything, no? Huh. Now students, we have to write about lipase, peptase and protease, okay? Pectinase and protease. Let's write that also. Okay. So lipase is used in detergents. Okay, I'm only writing that much. How it's used, I'm not writing that. Then proteases and pectinase. Okay, pectinase is used for clarifying fruit juices. So that's why don't drink bottled juice. Okay, don't drink bottled juice because they are clarified. That's not your fruit juice. Okay, they might write organic, but that's not organic. Please understand, nothing organic exists. Okay, all scam. Done? Okay, chalo, next one. Microbes in sewage treatment. It's a very, very simple treatment. Okay, before I read this, I will directly get into the sewage treatment, then I'll come back to this. Okay, then I'll come back to NCERT. So we start from here, students. So students, what is the sewage treatment? It's very simple, that all of our excreta, all the waste material that is being used has to be purified before even sending it to, or releasing it to any river or sea, whatever, lake or anything. This was the technique before. Now what happens, since water has to be recycled, water has to be used for other thing, we do a technique called as STP, that is sewage treatment plant, where all the sewage will be purified with the help of 2 steps called as primary and secondary, or you can call it as physical treatment and biological treatment. Physical treatment is where we can remove the debris. Now imagine in this glass of water, okay, in this glass of water, here there was a lot of sand and here water was there, or some floating debris, some plastic cover, some cigarettes, some things were there. Now I can remove this physically. I can remove the plastic cover, cigarette physically using a what do you call that, sieve. Okay, I can remove that. Now some of them have got sedimented. So in my primary step, right, in my primary treatment, what is going to happen? They will basically have 2 main steps. The primary treatment It's a very simple, okay, in this chapter only this, some students tell, ma'am, this also is difficult, but it's not. Okay, primary treatment, students, first one is where we do sequential filtration. Okay, sequential filtration. Now, what is sequential filtration? That means I filter out whatever is physically possible for me to filter out. Okay, sequential filtration, where I remove all the debris, right? Okay, done. Now, second step is going to be sedimentation in the primary step itself. Okay, the second step is going to be sedimentation. Sedimentation. Now, in sedimentation, what is going to happen? Whatever I cannot physically remove, right, I will make sure that it settles. Okay, thank you, Madhumati. Okay, I will make sure that they settle and then I remove them. Okay, so look here. Primary treatment, these treatment steps basically involve physical removal of particles, be it large, be it small, anything. Anything can be there in the water, no? Okay. Now, from the sewage through filtration and sedimentation, these are removed in stages. Initially, floating debris is removed by sequential filtration. So, floating debris, I am going to mainly focus to remove it with the help of sequential filtration. Whatever is lightweight, everything will be floating on the top, right? Floating on the top of the water. So, that I will remove it with the help of sequential filtration. Any doubts so far? Okay, this is the first one. Let's add some leaf, okay, some plastic covers, okay. I don't know what is that, some cigarettes, okay, all these are the ones which I'll be able to remove physically, okay. Now, next one is, now look here, initial floating debris is removed by, then the grit or the soil and the small soil, that is pebbles, no, that will automatically get settled down. So, here what am I basically going to remove? All the grit. Grit is too difficult word, then just write soil and pebbles, okay. Soil and small pebbles. Students, only these things will be there in the water? No, right? The main thing is your excreta, that is organic material which cannot be seen with the naked eye. It would be a mixed, right? That also has to be removed. That will go for your secondary treatment, okay? So, these things will be removed. Whatever is possibly visible to us will be removed this way. That is called as a physical treatment. So, same thing. Let's draw some water. Now, in the bottom, there will be some sedimentations. Okay, some sediments will be there. These are your sediments. Okay. Now, students, this water, no, after I removed the whatever was on the top, I removed those debris. In the between that is there, that will be called as my effluent. Primary effluent. Is this clear? Any doubts so far? Please ask me. STP is sewage treatment plant. Okay, sewage treatment plant. So, this water, right, this water where the sediment is there, everything is removed out, that will be called as my primary effluent. This primary effluent is going to the next step. Okay, primary effluent. Now, let's see the next step, that is your secondary treatment. Secondary treatment, what is going to happen? Or biological treatment, the primary effluent was that water, is passed into a large aeration tank. Okay, large aeration tank. So, let's draw one tank here. This is one large aeration tank, students. Okay, this is the tank. That primary effluent, I have transferred it here. Now, where it is constantly agitated mechanically and air is pumped into it. So, aeration tank means obviously air should come in, no? So, this is the through which the air is going to come in. Okay, air is going to come in. So, let's write here, this is the aeration tank. In the aeration tank, what is there? This flow, if you understand, then everything is easy. Okay, primary effluent is there. Primary effluent is there. Now, look here, what is happening? Continuous mixing is happening. Okay, mechanical mixing is happening at the same way with the air also is pumped in. This allows vigorous growth of useful aerobic microbes. What do microbes want? They need oxygen. Now we are continuously pumping in air, so obviously the microbes will start growing on them because they also have organic material inside this water which they will be consuming. So this will be called as flocks. What will be this called as, students? Flocks. This aerobic bacteria, right? This aerobic bacteria will be called as flocks. Done? So far good? Yeah, I want all of you to wait here for 1 minute. I forgot my water bottle, so I'll just get some water. Stay back. Don't go anywhere. Till that, mark the attendance, all of you. Yes. So fast ad came, no? Yes, students, they have increased the ad. I see it. Why, Divya? Why are you not listening? Okay, so look here, students. So from the primary, I took the primary effluent and I added into aeration tank where continuously air is coming and mixing is happening. Okay, mechanical mixing is happening. Because continuously air is coming in, the bacteria will feel happy and start growing. Which bacteria? Aerobic bacteria. And that, what to tell, group of bacteria, right? We call it flocks. Okay, so aerobic bacteria into flocks, that is masses of bacteria associated with fungal filaments to form mesh like structure. So you can basically call that mesh as flock. Okay, while growing, these microbes consume large part of organic matter in the effluent. So that's why I told you whatever was visible to us was removed in the primary treatment. Now many organic, sewage means what? Our kitchen waste, our washroom, the toilet waste, everything is mixed up in this. In this, the maximum thing is organic matter. That organic matter will be consumed by this aerobic bacteria. Okay, this significantly reduces the BOD, that is, students, biochemical oxygen demand of the effluent. Always remember, DO means dissolved oxygen. Okay, DO means dissolved oxygen. This is the oxygen that imagine there is a river in that DO. Okay, DO is there. Dissolved oxygen is the one that is required for all the aquatic animals, say the fishes, say the planktons, all of them want dissolved oxygen. Okay, but if algae is there in that, then this dissolved oxygen automatically reduces because they will start consuming it. Now at the same time, BOD, okay, biochemical oxygen demand is more. Okay, that means what? Here what is going to happen? See, this significantly reduces the BOD. So as the BOD reduces, we understand that the organic matter also in the water has reduced. If BOD is more, then we have to understand, oh wait a minute, lot of organic matter is there, this aerobic bacteria is asking for more, right? More organic matters are there. So BOD automatically reduces means it's a good thing. That means when BOD reduces, automatically dissolved oxygen increases. The good amount of oxygen, no, which the other organism wants, they will be able to consume that. Now effluent reduces. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. Don't break your head, just know BOD. Okay, don't break your head for understanding the definition, right? Because definition won't come. They will just ask you the BOD will decrease or increase during the secondary treatment. Such questions can very rarely come, but yeah. So sewage water is treated till the BOD is reduced. That's what I'm telling. If BOD does not reduce, that means please do understand that the water has still organic matter and the bacteria is still feeding on them. If the BOD keeps decreasing, that means water is getting purified. Okay. If dissolved oxygen is more, that means water is purified only. Water is there, good amount of oxygen is there in the water. Okay. Now, the BOD test measures the rate of uptake of oxygen by the microorganism in a sample of water. Thus, indirectly, BOD is the measure of purity of the water. Okay. Is the purity of the water. The greater the BOD of wastewater, more it is the polluting potential. Okay. Yes, more is the polluting potential. Clear? So, students, this one, yeah, okay. Now, so now the BOD we tested, we understood that the BOD is reducing. That means the bacteria are actually doing the job. Now, look here, the effluent is then passed into a settling tank. So, two tanks are there, aeration tank, settling tank. First, all the sedimentation happened. From there, we took the primary effluent, put it into the aeration tank. We gave enough air. Flocks were grown. That started consuming the BOD. BOD kept decreasing. Now, from there, that effluent, I have transferred it into the settling tank. Okay, this is the last. We are coming to the final step. Now, the settling tank, where the bacterial flocks are allowed to sediment. Okay, the bacteria, no, we are allowing them to sediment. This sediment is called as activated sludge. So, students, this is the tank. Okay. This is the settling tank. In the settling tank, we have the water. Okay, water is there. Now, at the bottom, okay, at the bottom, we have the bacteria. Okay, the aerobic bacteria, which we called as the flocks. Okay, now this will become an activated sludge. Okay, this bacteria, this is your flock, right? This is your aerobic bacteria. Okay, aerobic bacteria. This will be called as the activated sludge. Okay, activated sludge. Activated sludge. This is the only, yes, correct, Sony, correct. Okay, so look here. Activated sludge is pumped back into the, so a small part of the activated sludge. So, student, this one, now imagine I used something that would harm me indirectly in the tank. Now, it's my responsibility to make sure that that is also removed, right? Imagine this water has to go for another purpose, for drinking. Then it has to go for more purification. But imagine this water has to be sent into a river or sea. There are aquatic animals. I have used aerobic bacteria. Now, it's my job to kill this aerobic bacteria. So, for that, I will use an anaerobic digester, right? Obviously, they will consume it. Now, but some amount of it, like how you had your inoculum in curd, no? Like that, some amount of the activated sludge, I will use it back into the first step. As an inoculum. Imagine bacteria I already had. I'll use some of them back into it. Okay. So, look here. A small amount of the activated sludge is pumped back into the aeration tank. We are now in the settling tank. This is settling tank. From the settling tank, okay, settling tank. I'll draw the whole thing. Okay, I'll write the whole thing once again. From the settling tank, I'll send some amount of the activated sludge to aeration tank. Okay, aeration tank. Now, rest of them, what am I going to do? I am going to take the rest of the sludge is pumped into a large tank called as the anaerobic sludge digester. Okay, I have to finish that bacteria. I don't need them anymore in the water because they can also cause me problem. So, I will digest it, right? Not me, students, this one. Anaerobic sludge digester will be there. Here, other kinds of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge. Remember, they had fungus filaments. Okay, that only form the mesh. We cannot leave fungus in the water telling that, oh, this fellow digested all the BOD, so I'll leave him in the water. No, it is still harmful. So, I will use them or I'll consume them with the help of anaerobic sludge digester. Now, during this digestion, bacteria produces a mixture of gases such as methane, hydrogen sulfide and carbon dioxide. Now, some of you can ask me a question telling that, ma'am, okay, Yes, this is one shot. Okay. Then some of you can ask me a question, ma'am, what about this anaerobic bacteria? Students, anaerobic bacteria will automatically die when you leave them in an aerobic condition, right? Automatically they will die. So you don't have to take tension about that, but you have to take tension about the aerobic one. That's why we are using an anaerobic sludge digester, which will digest the flocks that we used. Some amount of the flocks went back to the aeration tank. Now look here. These gases form biogas because methane forms biogas and can be used as a source of energy and it is inflammable. The effluent from the secondary treatment plant is generally released into natural water bodies, but if you want to use it for drinking, then you have to do more purification. Okay, if you want to use it for drinking, then you have to do more purification. Got it? Very sad, Trisha. What do we do? Okay, what do we do? Okay, chalo. So let's write it, all of you. Ready? Start from the starting. I'll divide the board into two. Okay. PYQs we'll take it next class, Abhi, because okay, another teacher also has a class here, so I have to finish the class before that ma'am's class starts. Okay, so we will do the PYQs later. Don't worry. Okay, students, let's write primary here, secondary there. Okay, all of you start fast. You have to tell me. Then we'll finish this. Then we have only biogas and biocontrol and biofertilizers. Very easy. Now look here. Sewage treatment I'm dividing into two parts. Here is where the primary treatment is going to happen. Okay. Primary treatment. So primary treatment basically has two steps. What is the two steps? Sequential filtration. That will remove floating debris. Yes, floating debris. Okay. Okay, fast. It's a little more faster. Okay. Then we have sedimentation. Okay, sedimentation. This is going to do what, students? It's going to remove grit, or I would say soil and pebbles. Okay, soil and pebbles. Now I am drawing one diagram so that we understand which is the primary effluent. Okay. No tanks are spoken in the primary treatment, so please keep in mind that. Okay, no tanks are spoken in primary treatment, so please keep a note of that. Okay, here let's say the top portion is the filtrate. Filtrate means whatever unnecessary things are there that we are going to use, okay, as filtrate, and down we will have your sediments. Whole board is moving. This is an ASMR video. Okay, yes. So here this portion, okay, this is going to be my primary effluent. Okay. Now this primary effluent is getting into the secondary treatment. Clear? Image is clear? I should have used another color. Okay, primary effluent. Fine. This primary effluent is going into the secondary treatment. So then secondary treatment or biological treatment, okay? If you see the word biological treatment, don't get confused. Secondary treatment. Okay, this primary effluent, okay, this primary effluent I am making it enter. It is entering into what tank? Aeration tank. Okay, it's getting into the, I am drawing the tank closed. Okay, this is the aeration tank. Okay, done. Okay, very good, very good. Okay, this is the aeration tank. In this, what is happening? Continuously air is pumped in. Okay, air is pumped in, and this will lead to growth of aerobic bacteria. Okay, growth of aerobic bacteria on the, and this aerobic bacteria will be called as flocks. Okay, they are called as flocks because they are a mesh, right? They are a mesh of bacteria with fungus filaments. Okay, mesh like. Now what is going to happen, students? What is the next step after this? Is the colors visible? Huh? Everything became white, blue. Just tell me the next step. By then I'll just change the color of this. Yes, quickly in the chats. Yes. Yes, done. Okay, flocks are there, done. After flocks, what? Now, students, after mechanical mixing, this bacteria will start consuming, right? And because of this, the BOD will reduce automatically. This will show us that the organic matter also has automatically reduced. Okay, this will show that the organic matter has also automatically reduced. Okay, now this one, right? This will become the secondary effluent, if you want, we can call, but let's just keep it effluent. Okay, this effluent will be passed, or this whole thing will be passed into the settling tank. Okay, which will be passed into the settling tank. Okay, this whole thing will go into the settling tanks. In the settling tank, what will happen, students? In the settling tank, some amount of the sludge, right, some amount of this activated sludge will go back into the aeration tank, and the rest of them will go into, right, rest of the activated sludge. Activated sludge, that is your flocks, right, will go into what? Will go into anaerobic digester. Anaerobic sludge digester. Okay, sludge digester. On this, I am drawing some this one. Okay. Okay, some amount of this, right, some amount of this will go back into the aeration tank. That is some, I'll write it here. Some amount of activated sludge will go back into the aeration tank. Is this clear? Okay, done. All the colors are there. What, yeah? What are you people telling? Yeah, okay. Tanuja told something nicer. Yes, students. So, activated sludge is there. This will go into the anaerobic sludge digester. All of them will be digested. Now, this water, right, this one will be going into the, we can send them back into the rivers and seas. Okay, rivers and lakes, not seas, rivers and lakes. And if you want to further consume it, like you want to drink, okay, you want to drink it, then we have to go, okay, Okay, nice. We have to go further for rest of the purification. So from here, if you want to use it for drinking, then again further this will be there. Okay, clear? Clear? Okay, this is your sewage treatment, secondary treatment. So here I am going to write few things. Okay. So here students, activated sludge, okay, is your flocks itself, okay, and after the secondary treatment, okay, after secondary treatment we have to go for purification. Tertiary treatments will be there, okay, purification, to use the water for drinking. Clear? That's all. That's your secondary treatment. Okay, done. Okay, got it. So done. That's all. Okay, sewage treatment. Now students, one plan was made by the government which is called the Yamuna Ganga Action Plan. Okay, because you understand, no, lot of pollution happens in these places mainly. So the Ministry of Environment and Forest has initiated Ganga Action and Yamuna Action Plan to save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of STPs, okay, sewage treatment plants, okay, sewage treatment plants. It is proposed to build a large number of sewage treatment plants so that the only treated sewage may be discharged into the rivers. A visit to the sewage treatment plant situated in any place near you would be interesting and educating. Okay, so you can go and check any of the treatment plants. Now students, when I was teaching that, all of you would have noticed that they were telling that these people release some gases, right? What are the gases that they were releasing? They were releasing mainly methane. Okay, they were releasing H2S, hydrogen sulfide, okay, carbon dioxide. All these gases were released by these bacteria or your microbes. Now I can use these gases to form biogas. Now how do I form it? Very simple. See, there is a mixture here. No, you see the first tank here. This is the most important thing. This is the first tank here. Now this someone got it. Okay, so Sony got it. Okay. Students, you have to forgive, okay, forgive. It's when I have to talk like this. Okay, so this is the first tank. In this tank, what we are going to do is we are going to mix gobar and water, okay, gobar plus water. That means your cow dung, okay, cow dung plus water. Now, why am I using cow dung? Very simple. What does ruminant animals have? Methanogens in their gut. No, they have methanogens in their gut. This methanogens will be able to produce methane, and this methane gas can be used for production of biogas or for anything, any other purpose, right? Okay. So what is going to happen? This cow dung is rich in methanogens, or this ruminant animals, no, cattle, they have methanogens in their gut which will be able to form methane. So this methane will be used to form biogas. This methane will be used to form biogas. So this is why we are using cow dung. Okay, students, nothing is there. This cow dung plus water, right? We call this as the slurry. Such words I will not get confused. When words are come normal, that's when I get confused. Okay. When I tell slurry and all, no slippery will come. When I tell something else normally, gone. So this is slurry, that is gobar plus water or cow dung plus water. They will be kept here. Then there will be a pipeline to it. Now what is going to happen? This is a digester where the gases will be continuously mixed, and this will release the gases, that is your methane will be released. Now can you see here? So students, here there is one, let's say a gas holder, and there is one, imagine a plate is there, okay, a big plate is there. Just imagine like this, one big plate is there, and you're boiling something. Okay, let's imagine water is there and you're boiling it. Have you seen the plate will tremble like this? It will go up, come down, go up, come down. That is why, because gases are getting released. Okay, so same way, same thing will happen here. The digester will continuously digest this slurry and gases will be released. That gases will be contained or stored into a gas holder. okay, from the gas holder. Now imagine whatever the gases were released, okay, fine, but there will be some more left, no, some more left will be there, some mixing will be there, that we will be using it as manure. Okay, manure, that means you can use it as fertilizer, you can use it for, because so much, understand, this will have a lot of other waste materials as well, okay, so that we can use it, that will be called the sludge, okay, that we can use it as manure. So biogas will be formed, that will be taken here and it will be circulated in all villages, so they can use it. The rest, whatever the rest we got from this, no, that will be called as sludge, or you can tell that the rest will be used as manure. Okay, the rest will be used as manure. So whatever rest happened, that is called as sludge, and this we will be using as manure. Is this clear? Biogas is so easy, yaar, very easy. Okay, very easy, nothing is there. Okay, nothing is there. Nicely, people are having fun. Okay, have fun. Okay, next time you people stand and teach like this, that time I'll sit and laugh. Okay. Chalo. Some dancer is dancing. Okay, even I am getting same ad students. Okay. Yes. Next one, is this clear? A slurry is there, went into the digester, mixing happened, gas got, like let's say gas holder was there, okay, and then those gas, that is the biogas, is being supplied to everyone. Now whatever is rest will go as sludge, which will be used as fertilizers or manure. That's all is your biogas. Clear? Next is biocontrol agents. Okay, biocontrol agents. Now students, biocontrol means, see, when I am, let's say there's one spammer in the class. Now is there point of me scolding the whole crowd? No use of scolding the whole crowd, no, because the rest of the students are very loyal, they have come here for study. Because of one black sheep, should I just stop taking classes on YouTube? No, I cannot do that, right? Same way, like you would have heard your teachers also tell, because of this one black sheep, why should I spoil my name or whatsoever? Same way, if you are targeting one insect, why are you killing all other insects? Maybe some of them are the good ones, but why are you killing all of them? Okay, so same way, biocontrol agents means they will target only one set of people. Now imagine BT cotton, Bacillus thuringiensis, problem is there. Okay, Bacillus BT cotton, I want to attack only the larvae of that. I will make a biocontrol, or I'll make a biofertilizer or whatever pesticide in such a way that it will only target that larvae. Even any other insect comes, it will not target them. So that was found because of genetic engineering. Okay, so look here, biocontrol refers to the use of biological methods for controlling plant diseases and pests. And see, this is a control technique. It's not going to completely kill, okay, completely kill the organism. It is the control technique. So in modern society, these problems have been tackled, increasing by the use of chemicals. Now for us, all the technique has only one answer, that is use as much as chemicals possible. No, something is wrong, okay, put chemicals. Insect, cockroaches there, put hit everywhere, hit. Okay, even if a person die because of the hit, doesn't matter, put hit everywhere. Mosquito is there, black hit has come, okay, black hit everywhere. So that means for us, everything has solution at our fingertips. Because of that, we keep using chemicals so much that, students, you won't believe, I was just recently just checking this organic curd, Greek yogurt and all that. So wherever, okay, the organic word was written, I was just thinking, and then I ordered one. And if you go to your grandparents' home and if they make curd, or even at home also if they make curd, within a day, the curd will start getting very bitter, or it will get very sour. Same way, this curd also, even though I kept it in the fridge, it got very sour. And I got very angry that, why did this happen? Okay, I bought it yesterday night, and how can it... But that is natural. Okay, that is natural. That means normally it should get a little more sour and bitter. But what happens, once we buy otherwise, right, from normal stores, that does not get anything. It will stay like that forever. Okay, it will stay like that. Even if you keep it for 10 days and take it later, it will be like that only. I feel they have put bleaching powder in that. What do you guys think? Okay, so let me know in the chats. So in modern society, these problems have been tackled by the use of chemicals, by the use of insecticides and pesticides. These chemicals are toxic and extremely harmful. We all know that. So this is just a story. Okay, now the actual topic is coming here. Okay. So you look here, biological control of pests and diseases. In agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemical. Now look here, a key belief of organic farmers is that biodiversity further, improves the health. Okay. Now, the more variety a landscape has, the more sustainable it is. The organic farm therefore works to create a system. I don't know why they're telling stories, but okay, now look here. Contrary to that, we have a conventional farming practices which often uses chemical methods to kill both useful and the ones which are good also. Okay. Yes. What do you guys think? Have you ever tasted an organic food and then gone back to the artificial chemical one, telling that no, actually my body loves chemicals. Okay, I am that person. So yes. Okay, harmful life forms. So, students, all this I'm going to skip. Okay, I'm going to start from full story. This is full story. Okay, this is just to tell us about how important it is to use biocontrol pesticides and weedicides instead of using chemicals, because what happens is chemicals, when you're using chemicals, students, they are not specific, right? They're not specific. They are like, okay, I am a chemical, I will kill everything. So it will kill everything. Good, bad, why you have come here, okay, doesn't matter, kills everything. But biocontrol agents are not like that. Okay. Biocontrol agents are the ones who will be very, very specific. So they're talking about that. Now look here. The very familiar beetle with red and black markings, the ladybird, and the dragonflies are used to get rid of aphids and mosquitoes. So if you want to kill mosquitoes, put dragonfly in your house. Okay, and if you want to kill aphids, put ladybird in your house. That's the solution. Okay, don't tell it to anyone, top secret. So an example of microbial biocontrol agents that can be introduced in order to control butterfly caterpillars are your Bacillus thuringiensis, which you will study in detail in biotechnology. Got it? So here they're trying to say that why are you trying to put chemicals for everything? Why are you using HIT for everything? If there are mosquitoes, put dragonfly. Okay. So this is one good example. Ladybird kills aphids. Okay. And dragonflies kills mosquitoes. Clear? Mosquitoes. Now, students, this BT cotton, right, Bacillus thuringiensis, is a very amazing technique. Why? Okay. Why? Because when the larvae consumes, right, immediately this gets activated in the stomach of the larvae. Why? Because the larvae has an alkaline pH in the stomach. Why? When we consume the same thing which has been sprayed with Bacillus thuringiensis, when we consume, let's say, tomato or lady's finger or anything of that sort, we do not get harmed. Why? Because we have an acidic pH in our stomach. The larvae's pH is alkaline. So only when a larvae comes and consumes it, the larvae will get harmed. Okay. So here they are telling that these are available in sachets as dried spores which are mixed with water and sprayed on vulnerable plants such as brassicaceae and fruit trees. Whereas these are eaten by insect larvae. So in the gut of the larvae, this is all that you have to remember. Okay. In the gut of the larvae, the toxin is released and the larvae gets killed. Now look here, the bacterial disease will kill the caterpillars but leave other insects unharmed. So this is the technique called biocontrol. So in case a question comes, what is the advantage of biocontrol agents? This is the main advantage, that they are very specific. They will only kill what they are assigned to kill. Okay, they will not kill everything and anything. Okay, it's not GTYCT, no. So because of the development of methods of genetic engineering in the last decade or so, the scientists have introduced Bacillus thuringiensis toxin genes into the plants. So that's all. You can just tell the larvae is getting killed mainly because the larvae has alkaline pH and it's not affecting the humans because we have acidic pH. Okay, in our stomach. Okay, alkaline pH in the gut of larvae. Clear? All of you? Okay, fine. Sony has got panic because of bleaching powder. Okay, now a biological control being developed for the use in the treatment of plant disease is fungus trichoderma. Okay, This Trichoderma species are free-living fungi that are very common in the root ecosystem. They are effective biocontrol agents of several plant pathogens. So, they are telling that Trichoderma species are free-living. It's a fungus, but they are useful. Okay, that are very common in the root ecosystem. They are effective biocontrol agents. Okay, now same way baculovirus are also pathogens that attack insects and other arthropods. The majority of the baculovirus used as biocontrol agents are in genus Nucleopolyhedro. Are you all following me? So students, here what are they telling? They are telling that Trichoderma is there, which is a free living fungus which actually helps many plants by living in the root system. Okay, Trichoderma. Here they are talking about baculovirus. Okay, baculovirus which can actually kill many or attack many insects. Attack insects and arthropods. Okay, insects and arthropods are used as biological control agents, are in the genes of Nucleopolyhedrovirus. These viruses are excellent candidates for species specific. So, these are present where? In the Nucleohedrovirus species. I mean genus. Okay, right? Genus, no? Genus, and they are very species specific. God. Okay, narrow spectrum insecticidal application. Students, this word you have to remember. Narrow spectrum means if I am targeting only one student in the class, my target is on that student only. I am not changing my target to any other student. I am only targeting that student, or I am only complementing that student. That means what? I am biased, no? They tell, no? That means I have a very narrow spectrum. I am only focusing on that one point of view, that is the narrow spectrum. So, same way this one also, Nucleopolyhedrous virus is very narrow spectrum. They are very specific. Okay, why they have come here? They will do only that job. Okay, insecticidal application, they have been shown to have no negative impact on the plants or the mammals that we are spraying them. Okay, birds, fishes, or not even to the non-target insects. This is especially desirable when beneficial insects have been considered to aid in an overall integrated pest management program, or when an ecologically sensitive areas is being treated. Students, when I taught you this, no, one thing, biogas, you have two full forms, right? All of you open NCERT and just tell me, you have two full forms, no? Khadi village, okay, IARI and KVIC. Yes or no? Just check and tell me. Khadi Village Industrial Commission, Ganga Yamuna Action Plan, I told you. Indian Agriculture Research Institute, okay, Indian Agriculture Research Institute, and Khadi Village Indian Commission. Yes, these two names you have to remember, full form, okay, Industrial Commission. These two full form they might ask you, okay. Yes, just check and tell me, is there, right? I think, yeah, okay, Indian, I forgot to mention it here, Agricultural Research Institute. Yes, okay, Khadi Village Industries Commission, okay, Khadi, these people only help this bio, okay, Khadi Village Industries Commission. This happens mainly in the village areas, okay, biogas production, because there's a lot of cattles there. Their main, some people's main, what is that? Earning is by cattles, okay, yes. So students, this was clear. So this integrated pest management program, or when an ecological, so here you have to know some full forms like IPM, that's when I remember that, IARA and KVIC, all these names you have to remember. Same way Ganga Yamuna Action Plan. Next one, See, we have now realized that there are problems associated with the overuse of chemical fertilizers and there is large pressure to switch to organic farming. The use of biofertilizers. So, now we finished biocontrol and next we are going to biofertilizers, okay. I'll write this in GM Express here. Once I finish this 2 paragraph, I'll write it here, okay. Biofertilizers are organisms that enrich the nutrient quality of the soil. The main source of biofertilizers are bacteria, fungi and cyanobacteria. You have studied about the nodules of root of leguminous plants formed by the symbiotic association of rhizobium. All of you know this, no? They help in fixing the nitrogen and this is also done to increase the fertility of the soil. So, if you ask farmers, you can see some leguminous plants kept between paddy. Okay, that is because they believe that yes, this actually increases the fertility of the soil. Okay, these bacteria fix atmospheric nitrogen into the organic forms which are used in the plants as nutrients. See, other bacteria can fix atmospheric nitrogen while free living in the soil, like example azospirillum and azotobacter. These 2 examples also very important. I'll make you write this, thus enriching the nitrogen content. So, these people can fix nitrogen without being associated with anyone, free living. Okay, single, happy, free living people. Next one. Here students, mycorrhiza they are talking about. All of you know mycorrhiza is present in higher plants. The one that is present is glomus. Okay, the genus is going to be glomus from mycorrhiza. The fungal symbiont in these association absorb phosphorus from the soil and passes it to the plants. Okay. Plants having such associations show other benefits also, such as resistant to root-borne pathogens. See, no other pathogen will be able to come and attach themselves because mycorrhiza, this association is already there. So, this way plants not only get phosphorus, but they also get, they can avoid some root-borne diseases, tolerance to salinity, that is soil, salt content and water. Okay, drought condition and an overall increase in the plant growth. See, this mainly happens in tall trees. Okay, tall trees. Okay, these trees will be, students, they will obviously lack something because they're so tall, they have to get the whole thing past. So, that time this kind of symbiotic associations are working. Okay. Now, here cyanobacteria, 3 things they spoke about, right? First thing they spoke about was here about your bacteria, fungi. See here, bacteria, fungi and cyanobacteria. In bacteria, here we saw some acetobacter. Here they are talking about fungi. Here they are talking about cyanobacteria. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial, and they can fix nitrogen, okay, atmospheric nitrogen. Anabenna, nostoc, oscillatoria. See, among this, oscillatoria does not have your heterosis cells, the ones that will fix nitrogen. If you read a little about it, you will get to know. Before it was believed that they had, but recent studies have proved that they do not have it, okay. So, oscillatoria does not have heterosis, that is nitrogen fixing cells, which I taught you in biological classification. In paddy fields, cyanobacteria serve as an important biofertilizer. Okay, blue-green algae also add organic matter to the soil and increases fertility. Currently in our country, a number of biofertilizers are available commercially in the market and farmers use them regularly. Students, same way your single cell spirulina, okay, protein, single cell protein spirulina also is used. You know that people who go for aerospace and all that, they have this spirulina because that consumes, like, because they will not have all the nutritious food, no, so they use that. So many things like that are there, right? Many things. Okay, shall we write it? Yes, ready? Chalo. Sony, let them do their job, yaar. They're paid money for that. Okay, yes. So what is it? We are going to talk only about biocontrol agents. Biogas I did. So biocontrol and biofertilizers, okay? Okay, tell me the different biocontrol. First we have your ladybird. We'll start from the basic, okay? Ladybird. Ladybird actually helps in killing aphids. Okay, getting rid of aphids. Then we have dragonflies. Okay, that will actually help from mosquitoes. Yes, mosquitoes. Got it? Then students, next one, tell fast. Yeah, exactly. So irresponsible. They'll keep coming now, don't worry. Just be calm. Okay? This is the message that I put on Telegram. Female teachers always get attacked. Yes, dragonflies done, mosquitoes done, ladybird done. What are the other biocontrol agents? Okay, most common one is Bt cotton, that is Bacillus thuringiensis. Okay, Bacillus thuringiensis. Yes, correct. Bacillus thuringiensis. Got it? They will be getting them in sachets, and you can mix it with water, and these people will attack the butterfly caterpillars. Okay, butterfly caterpillars. Got it? Next is trichoderma. Trichoderma helps in root ecosystem, that is to improve the root ecosystem. Okay, students, then baculovirus. Baculovirus, yes. If I met them, definitely. Okay, baculovirus, nucleohedrovirus genus. Okay, these are species specific. Done? Yes. Tanuja is the only calm one in the class. Tanuja is like, it's okay, guys. Okay. Yes. Nucleohedrovirus, species specific, narrow species. Okay, done. Then, students, biocontrol done, biofertilizers. Bio. Okay, biofertilizers. Yes. Okay, so biofertilizers is gonna be your, you can, three, okay, bacteria, fungi and cyanobacteria. Okay, bacteria you have two only. I don't like this color. Okay, I'll have to. Sony is gonna kill them at this point. Don't worry. Okay, they're gonna keep coming. Now only they got to know that my class is happening. Happens. Okay. Now students, bacteria. I'm gonna write few names. Okay, they are free living. That is Azobacter and Azospirillum. Okay, they are free living. Got it? And fungi, you have Rhizobium, Okay, rhizobium is there, mycorrhiza is there. Mycorrhiza, I'm writing glomus, okay. Okay, mycorrhiza. Done? Chalo. Okay, yes. That's all, okay. This is cyanobacteria. You can write your blue-green algae, okay. Blue-green algae. And most of the cyanobacteria, okay. Again, you can write Anabaena. Oscillatoria. Nostoc. I thought my tongue twist thing will tell Annabella, but thank God it did not. Okay. The board is not dirty. It's a theme, yeah, kid. Okay, cyanobacteria, BGA, all this nitrogen fixation, okay. Nitrogen fixation, they could be free living or they could be in association, okay. Nitrogen fixation. Here also, here this fellow will do phosphorus fixation. Since phosphorus is PO4, phosphorus fixation, okay. Yes. Very good. Okay, we are done with microbes chapter. Chapter is over. Chapter is over. So we have written GM Express in between. Okay, see, biogas we wrote, sewage we wrote, then we wrote the most important slide, and then we wrote household. Okay, so everything we have written in between. So everything is there. Okay, four slides, full thing is there, and full NCERT is there. So students, thank you so much for joining. I hope you really enjoyed the session. We'll be doing PYQs very soon. Okay, we'll do a separate PYQ session itself where I'll be doing PYQs of all the chapters that I've done. So do not worry, tomorrow also we'll be doing one chapter. So be there tomorrow. And I hope the session was really helpful. And as I told you, do not forget to give me your Guru Dakshina. That's with your likes and comments. Do not fail to comment, students. It might take you some seconds, but it is going to help a lot. So definitely comment and let me know if the session was fun and happy and did you all like the session. Thank you so much for joining. I hope all of you loved it, and all these notes will be available on Vedantu NEET English Telegram. And that's it. Okay, if you want to join my Telegram, it's Biology by Gopika. Do not forget to comment in the comment section, not right now. Okay, thank you so much. Tata, bye bye. See you all. Students, I have a surprise. I have a surprise that I forgot to tell you people. You know what? I know some of you are regretting not joining the DNA batch. The offer is extended for 2 more days. The offer is extended for 2 more days. That is, you guys can still take your DNA batch for just 999. So do not wait anymore. I know some of you are feeling bad that, oh God, yesterday the offer got over. Now I got to know. 2 more days the offer will be extended. So do not go anywhere. Quickly go to the link, DNA batches there. Click on the link and join. And right now you will get the course again for 1000 rupees. So all of you can check. Okay. Yes, this is what I wanted to tell you. It was a surprise. Okay. So I hope all of you will do that. So thank you. Bye-bye.